1. What is the principle of gram staining? Answer. Gram staining is based on the ability of bacterial cell walls to retain crystal violet dye during solvent treatment. Gram-positive bacteria retain the dye because of their thick peptidoglycan layer, while gram-negative bacteria lose it due to their thin peptidoglycan layer and outer lipid membrane, taking up the counterstain, safranin. 2. List the steps of gram staining in the correct sequence. Answer. Application of crystal violet, primary stain. Application of iodine, mordant. Decolorization with alcohol or acetone. Counterstaining with safranin. 3. What is the purpose of using a mordant in staining procedures? Answer. A mordant, like iodine, fixes the primary stain onto the cell or tissue by forming a complex that enhances staining intensity and prevents the dye from being washed out. 4. What is the Zeil-Nielsen staining method used for? Answer. The Zeil-Nielsen stain is used to detect acid-fast organisms, such as Mycobacterium tuberculosis. Acid-fast bacteria retain the red dye carbylfusin even after decolorization with acid alcohol. 5. What is the counterstain used in the Zeil-Nielsen staining procedure? Answer. The counterstain is methylene blue or sometimes malachite green. 6. Explain the difference between simple and differential staining. Answer. Simple staining uses a single dye to color cells, e.g., methylene blue, and is used to observe basic morphology. Differential staining uses multiple dyes to distinguish between different types of organisms or cell components, e.g., gram staining, Zeil-Nielsen staining. 7. What are the steps of the hematoxylin and eosin, H&E, staining process? Answer. Deparaffinization, remove paraffin with xylene. Rehydration, pass through grated alcohols to water. Stain with hematoxylin to color nuclei. Rinse in water and use acid alcohol for differentiation. Bluing, neutralize with alkaline solution. Stain with eosin for cytoplasm. Dehydrate, clear, and mount. 8. What staining method is used to detect fungal infections? Answer. The periodic acid Schiff, PAS, stain is used to detect fungal infections by staining carbohydrates in fungal cell walls magenta. 9. What is the purpose of decolorization in gram staining? Answer. Decolorization with alcohol or acetone differentiates between gram-positive and gram-negative bacteria by removing the primary stain, crystal violet, from gram-negative cells due to their thinner peptidoglycan layer. 10. What stain is used to identify malaria parasites in blood smears? Answer. The Giemsa stain is used for identifying malaria parasites in blood smears, staining the parasite's cytoplasm pale blue and chromatin deep red.